Hello, my name is Thomas Renne. I'm a bioinformatics PhD student in the lab of Dr. Jacquemont. It's a lab focusing on decoding and quantifying the effect size of gene-disrupting variants on psychiatric disorders. So, I present today my work to create a polygenic risk score estimating any rare variant effect on cognition. CNVs are defined as genomic deletion or duplication larger than 1000 base pair. As represented here, a deletion leads to a single copy of the region A, which decreases the expression of the gene encompassed in this region. A duplication leads to 3 copies of this region, which increase the expression of encompassed genes. In the lab, we developed a first model quantifying the effect size of any gene on cognitive ability based on the fitness score of gene encompassed. These models show that gene intolerance to the loss of function will decrease the IQ for up to 5 IQ points for deletion and 2 points for duplication, while CNV of tolerant genes won't have effects on cognitive ability. However, this model uses only fitness score without taking the biological characteristic diversity in account. So, my hypothesis it is possible to develop a regression model to estimate the gene effect on cognitive ability using fitness score and biological functions. To answer this question, I'll begin by identifying if gene function has significant information to models. Then, I'll merge both fitness and functional annotation information into one model. To answer these questions, I use 250,000 individuals coming from general population and 6,000 from autistic cohorts. If you want more details, you could check the table presented here. In total, our individuals carry 460,000 genes, so I developed a model to compute the effect size of a specific gene list on IQ like presented in Model 1. The gene sets of interest are described in the table. Gene sets are a list of genes overexpressed in specific cortical layers or brain cell types. Then, I developed a more complex model based on the Model 1 with a stratification of genes based on their haploinsufficiency, the LUF score, where the genes are divided into 3 categories: the intolerant one, the intermediate one, and the tolerant. Here, I present a first result on a specific gene list. We have the results for deletion and duplication, and the y-axis is the effect size on IQ, and the x-axis the different gene categories. The black point corresponds to the first model, and the colored points to the second. And finally, the gray line corresponds to the naive model using only the LUF score. So, we can see that genes overexpressed in fetal excitatory neurons have similar effect sizes than intolerant genes, and when we stratify this data, we have even higher effect sizes for intolerant one, with a loss of almost 10 points of IQ by genes deleted. I replicate this analysis with all the gene sets presented before. The heat map summarizes the previous figure with deletion on the top part and duplication in the bottom. The effect size on cognition is defined by the color code: orange for positive effect sizes and purple for negative effect size. The dot size corresponding to the number of carriers, and the black edge is for significant effects. And again, we have the stratification with Model 1 as a whole and the Model 2 for the 3 other lines. We have a huge diversity of effect sizes across cell types, and we identify that genes overexpressed in glial cells such as astrocytes or microglia have effects on cognitive with a similar effect size than neurons. This matrix shows why developing a model using fitness score and biological characteristics is important, because we find a large diversity on effects across all cell types and love scores. To conclude this presentation, with our method, we identify that genes overexpressed in various cells have a broad range of effect sizes, and that cognition isn't only neurons related. To improve this analysis, we will replicate the results on all those gene sets and widening our analysis by computing effect sizes on genes overexpressed in non-brain organs and tissues and non-brain molecular functions using Go terms. Thank you for your attention.